Bismillahir Rahmanir Rahim. Today, the topic which we are going to discuss is transport across cell membrane. As we know, that the composition of intracellular fluid and the extracellular fluid which lies outside the cell is different. Hence, this difference of intracellular and extracellular fluid must be maintained. And the cell membrane has specific characteristics that it allows selectively some molecules to diffuse through or pass through it, while it does not allow other molecules to pass through it easily. The cell membrane is mainly composed of lipid bilayer, and there are integral proteins which lie inside the lipid bilayer, and from the lipid bilayer the lipid soluble substances can pass readily, while water soluble substances pass through the integral proteins which are present in the lipid bilayer. Now, this is the composition of extracellular fluid and intracellular fluid. Here you can see that the sodium concentration in the extracellular fluid is 142 milliequivalent per liter, while the concentration inside the cell is only 10 milliequivalent per liter. Similarly, the potassium concentration is much lower in the extracellular fluid as compared to the intracellular fluid. Similarly, the concentration of different ions is given in this table. The cell membrane ensures the concentration difference between extracellular fluid and intracellular fluid. This is the lipid bilayer structure of the cell membrane, and there are integral protein present within this lipid bilayer and allow the movement of water soluble substances through it. Cell membrane is selectively permeable barrier between the intracellular fluid and the extracellular fluid. Now, mode of transfer is that the molecules can easily move from one compartment to the other compartment by the process of simple diffusion or without involvement of any carrier protein. This Venn diagram that there is no carrier protein and molecules are moving from higher concentration towards the lower concentration. While with the help of the carrier protein, the molecules can even move from lower concentration towards the higher concentration by active transport, and the carrier protein are also used in the passive transport as well in some cases, as we will be studying later. Now, this is first of all, we are going to discuss diffusion. Diffusion is the movement of molecules from the higher concentration or movement of water molecules from higher concentration towards the lower concentration. This movement can also be of any of the gases like oxygen, carbon dioxide, etc., from higher concentration towards the lower concentration through the lipid bilayer, because oxygen and carbon dioxide is readily soluble and it can pass the lipid bilayer easily, and oxygen and carbon dioxide will readily cross the lipid bilayer and move from the higher concentration towards the lower concentration. Similarly, there are some channel proteins which are present in the cell membrane, and these channel proteins simply allow the diffusion of molecules from higher concentration towards the lower concentration. There is another example of facilitated diffusion, which is a bit different from that of the simple diffusion by the carrier protein. In this case, whenever the molecule binds with the carrier protein, there is a conformational change taking place in the carrier protein, and hence the molecule is moves from the higher concentration towards the lower concentration. This facilitated diffusion can also be gated, that there are some specific gates which when open allow the movement of ions from higher concentration to lower concentration, or it can also be voltage operated. Now rate of diffusion increases in direct proportion to the concentration difference between the inside of the between the extracellular fluid and the intracellular fluid. Greater is the difference between extracellular fluid and the intracellular fluid, greater will be the rate of diffusion. Lipid solubility, those molecules which are lipid soluble, for example, oxygen and carbon dioxide, can be delivered to interior of cell almost as if the cell membrane did not exist. Simple diffusion, the attributes of the simple diffusion through the protein channels is that there is selective permeability. The protein channels are selectively permeable to certain ions, and this selective permeability depends upon the size of the channel and also charge on the channel. The selective permeability of the protein channels for diffusion are based on the size of the channel as well as charge on the channel. For example, if the size of the channel is smaller as compared to the particle which has to pass through the cell membrane, then that particle cannot pass because of the larger size as compared to the size of the channel. Similarly, if inside of the channel is negatively charged, then negatively charged particles will be repelled and they cannot pass through that pore or the protein channel. Moreover, there is also gating of the protein channels. This gating can be voltage gating as well as the chemical gating. अब voltage gated का मतलब क्या है कि जैसे किसी बड़े मॉल में हम जाते हैं तो वहाँ पे जो ही दरवाजे के सामने पहुँचते हैं तो दरवाज़ा खुद ब खुद खुल जाता है, क्योंकि उसके जो फ्लोर है उसमें सेंसर्स लगे होते हैं, और वो सेंसर्स जो हैं वो क्या है कि वो स्टिमुलेट होते हैं और इलेक्ट्रिकली जो है वो दरवाज़ा खुल जाता है। इसी तरह जो प्रोटीन चैनल्स हैं उनके अंदर जब वोल्टेज चेंज आती है तो उस वोल्टेज चेंज की वजह से वो प्रोटीन चैनल्स ओपन हो जाते हैं, दे अलाउ द molecules to pass through it. Similarly, whenever a chemical binds with the protein channel, the molecules can easily pass through that channel, and such type of channels are also called as ligand gated channel. One of the example is that is acetylcholine gated channels. Now the gated channel, for example, in case of the sodium channels, the gates are present outside the cell membrane because the concentration of sodium is more outside the cells, and their gates are also present outside the cells, and whenever the gates open, sodium ion will enter into the channel and move along the concentration gradient inside the cell, while the potassium gates are located inside the cells, and whenever the gates open, the potassium ions moves from inside the cell to the outside, or from intracellular fluid to the extracellular fluid. Selectivity of these protein channel results from specific characteristics of the channel, such as its diameter, shape, and the nature of electrical charges and chemical bonds along its इनसाइड सरफेस। तो ये तीन चीज़ें जो हैं वो ज़्यादा इम्पॉर्टेंट हैं आयन चैनल की परमेबिलिटी को डिटर्मिन करने के लिए। वो एक इसका डायामीटर है, सेकेंडली शेप है, और थर्ड जो है जो इसके अंदर इलेक्ट्रिकल चार्जेस और केमिकल बॉन्ड्स जो है वो इनसाइड सरफेस पे प्रेजेंट होते हैं। तो ये चीज़ें डिटर्मिन करती हैं सेलेक्टिविटी ऑफ द प्रोटीन चैनल की। अब अगर सिलेक्टिविटी ऑफ चैनल्स की बात करें तो इनमें जो इम्पोर्टेंट चैनल है जैसे कि पोटेशियम चैनल है। अब पोटेशियम चैनल इज 1,000 times more readily permeable to passage of potassium than that of sodium ion. Although potassium is larger than the sodium ion, but the potassium channel is 1,000 times more readily permeable to the potassium ion. Why it is so? Because inside the potassium channel there are selectivity filters present, and these selectivity filters are made up of the carbonyl oxygens, and these carbonyl oxygen, they cause the dehydration of the potassium ions which are supposed to pass through the channels. Whenever the ions are present in the intracellular fluid and extracellular fluid, they are usually hydrated, that is, they are bound with the water molecules and their size is massively increased. But when they pass through their pores, there is dehydration of the ions, and the dehydrated ions become much smaller in size and can easily pass through the channel. So selectivity filters of the carbonyl oxygen which are present in the calcium channel, which are present in the potassium channel, they cause dehydration of potassium ion, and hence potassium can easily pass through the channel. The carbonyl oxygens are too far apart to enable them to interact closely with the smaller sodium ion. सोडियम आयंस के जो वाटर मॉलिक्यूल्स हैं उनके साथ ये इसलिए नहीं बाइंड कर सकते हैं कि दे आर लोकेटेड फार्दर अपार्ट, क्योंकि साइज ऑफ द सोडियम इज स्मॉलर, एंड ड्यू टू स्मॉलर सोडियम size they cannot bind with the water molecule of sodium and dehydrate the sodium easily. So sodium excluded by the selectivity filter by passing from the pore. Now, in case of sodium channel only allows the sodium ions to pass through it because of the selectivity filter which are present inside the sodium channel, which are strongly negatively charged amino acid residues and which only bind with the sodium ions. Because so in the sodium channel case there are strongly negatively charged amino acid residues which only allow binding with the sodium ions. Hence they pull the water from the sodium, and the dehydrated sodium ion can easily cross the sodium channel. Now next are the voltage gated channels. Voltage gated channels are also very important, and they are specially discussed when we are discussing the action potential. तो जो वोल्टेज गेटेड चैनल्स हैं उसमें मैंने पहले बताया कि जैसे बड़े बड़े मॉल्स में हम जाते हैं या हॉस्पिटल में इवन जाते हैं तो दरवाजे के सामने खड़े होते हैं तो वो डोर जो है वो ऑटोमेटिकली ओपन होता है बिकॉज इट इज इलेक्ट्रिकली आर वोल्टेज गेटेड ऑपरेटेड। इसी तरह जो ये वोल्टेज गेटेड चैनल्स हैं इनमें जब वोल्टेज चेंज आती है इन द एक्स्ट्रा सेलुलर फ्लूइड और द इंट्रा सेलुलर फ्लूइड, दिस voltage change causes the opening of the opening or the closure of the channels, and substances can pass through the from the extracellular fluid into the intracellular fluid or the vice versa. Membrane potential changes the conformation change in the receptors leading to opening and closing of the channel. तो जब membrane potential change होंगी तो there will be a conformational change in these receptors which leads to opening or closing of these channel. In case of resting membrane potential, when inside of the membrane is more negative, resting membrane potential में हमें पता है कि inside of the cell is more electronegative as compared to the outside, though the outside sodium gates remain tightly closed. तो रेस्टिंग मेम्ब्रेन पोटेंशियल के केस में जब इनसाइड जो है वो स्ट्रांगली नेगेटिवली चार्ज होती है तो जो सोडियम गेट्स है जो प्रेजेंट होते हैं आउटसाइड of the cell, they are tightly closed. Whenever the inside of the membrane loses its negative charge, these gates open suddenly and allow sodium to pass inward through the sodium pores, and it causes the depolarization phase of the action potential, because all of a sudden by the voltage gated channels will open and there will be flush of sodium ion inside the cell, which will cause the depolarization phase of the action potential. Then the voltage gated potassium channel. The potassium gates are on the intracellular ends of the potassium channel. जो potassium gates होते हैं, they are actually present on the intracellular end of the potassium channels, and they remain closed when inside of the membrane is negatively charged. They open when inside of cell membrane becomes positively charged, and it helps in repolarization phase of the action potential. So these are the voltage gated sodium and the voltage gated potassium channels, which we discuss especially in the resting in the action potential. There are also another type of channel which are called as ligand gated channels. Some protein channel gates are opened by binding of the chemical substance. जब कोई substance आके bind करता है channel के साथ, तो this binding of substance with the channel causes a conformational change or change in the chemical bonding of the protein receptor and opens or closes it. One of the example is the acetylcholine receptor in nerve and muscles for transmission of nerve signals. When acetylcholine binds with this acetylcholine receptors, it opens up the channel, and the size of opening of channel is about 0.65 nanometer in diameter, and neutral or uncharged molecules or positive ions smaller than this diameter can pass through this acetylcholine receptor. Now, what is the facilitated diffusion? The facilitated diffusion, in case of simple diffusion, there was just passage of molecules from higher concentration to the lower concentration, and there was no conformational change taking place. While in case of the facilitated diffusion, when the molecule which is supposed to be transported binds with the carrier protein, it causes a conformational change in the molecule, and hence the molecule is transported from the higher concentration to the lower concentration. This there is the specificity of this facilitated diffusion in case of the carrier proteins, because there is a specific point in the carrier protein. So we were discussing the facilitated diffusion. तो फैसिलिटेटेड डिफियन के अंदर ये है कि देयर इज अ करेक्टरिस्टिक ऑफ द वी मैक्स और मैक्सिमम ट्रांसपोर्ट, बिकॉज द मॉलिक्यूल्स have to bind with the receptor protein, and there is a limit to which the molecule can bind with this receptor proteins. After that maximum binding of the molecule with the receptor protein, further transport cannot take place, because there will be no more binding of the molecules to be transported with the receptor protein. And then there is a time which takes place from binding to the release. Hence, after a maximum level of transport, further transport cannot take place in case of facilitated diffusion. So this is example of the maximum transport. In case of this facilitative diffusion, you can see that after a certain extent, there will be a linear curve, and there will be no more transport of molecules across the membrane. While in simple diffusion, there is corresponding increase in the whenever the concentration increases, the transport will be increasing correspondingly. The examples of the facilitated diffusion are glucose transport and the amino acid transport in the GIT. Factors which are affecting net diffusion. Factors which are affecting net diffusion, there are certain factors which affect the net diffusion. ये हमें पता होना चाहिए कि कौन कौन से फैक्टर्स जो हैं वो नेट डिफ्यूजन को अफेक्ट करते हैं। जैसे कि कंसंट्रेशन डिफरेंस है, कि कंसंट्रेशन डिफरेंस जितना ज़्यादा होगा उतनी जो है वो नेट डिफ्यूजन ज़्यादा होगी। टेम्परेचर का इफेक्ट है, टेम्परेचर जितना ज़्यादा होगा उतनी डिफ्यूजन ज़्यादा होगी। सरफेस एरिया जितना ज़्यादा होगा उतनी डिफ्यूजन ज़्यादा होगी। मैस ऑफ डिफ्यूजिंग सब्सटेंस या कंसंट्रेशन या मैस, अगर मैस बहुत ज़्यादा होगा तो डिफ्यूजन उतनी कम होगी। डिफ्यूजन डिस्टेंस जितना ज़्यादा होगा उतनी डिफ्यूजन जो है वो कम होगी। मेम्ब्रेन एक्शन, इलेक्ट्रिकल पोटेंशियल और प्रेशर डिफरेंस जो हैं दे आल्सो अफेक्ट द नेट डिफ्यूजन। पैसिव डिफ्यूजन, passive diffusion or osmosis is the simple diffusion of water molecules across a semi-permeable membrane. Osmotic pressure क्या होता है कि whenever water molecules are moving across a semi-permeable membrane, there is pressure developing at one side, and that pressure will stop the further movement of water molecules. So the pressure required to stop osmosis is called as osmotic pressure. And now what are osmoles? ये बायोकेमिस्ट्री की चीज़ें हैं, लेकिन जस्ट हम इसको एक ओवरव्यू दे देते हैं कि osmoles are the number of osmotically active particles in a solution. ठीक है, नंबर ऑफ ऑस्मोटिकली एक्टिव पार्टिकल्स इन अ सॉल्यूशन। तो जो ब्रेकेबल पार्टिकल्स हैं उनकी ऑस्मोलैरिटी ज़्यादा होती है। जैसे NaCl है तो वन मोल ऑफ NaCl इज इक्वल टू 2 osmoles, while 1 mole of glucose is equal to 1 osmole. तो osmolarity is defined as number of osmoles per liter of solution. इसी तरह osmolality जो है it is defined as number of osmoles per kg of the solvent, per kilogram of the solvent. So ESF, extracellular fluid osmolarity, is mainly due to sodium ion, and it is 281 milliosmol per liter or 300 milliosmol per liter. Now, these are some terms which are usually used. What is hypertonic, hypotonic, isosomatic, hyperosmotic, and hypoosmotic solutions. तो ये terms आप यहाँ से slide से पढ़ लेंगे। तो कि हाइपोटोनिक सोल्यूशन क्या होता है? हाइपोटोनिक सोल्यूशन नॉर्मली उस सोल्यूशन को कहते हैं कि जो जिसके अंदर कंसंट्रेशन ऑफ द सॉल्वेंट इज मोर देन 300 मिली ऑस्मोल्स पर लीटर। और आइसोसोमोटिक सोल्यूशन वो है क्या? सोल्यूशन कंटेनिंग 300 मिली ऑस्मोल्स पर लीटर ऑफ द नॉन पेनिट्रेटिंग सोल्यूट्स रिगार्डलेस ऑफ द कंसंट्रेशन ऑफ मेम्ब्रेन पेनिट्रेटिंग सोल्यूट्स दैट मे बी प्रेजेंट। ठीक है, तो ये मेनली बायोकेमिस्ट्री की चीज़ें हैं, लेकिन ये है कि ये आपको पता होना चाहिए कि व्हाट इज आइसोटोनिक सॉल्यूशन, व्हाट इज हाइपरटोनिक एंड व्हाट इज हाइपोटोनिक सॉल्यूशन। इसकी इम्पोर्टेंस जो है वो क्या है मेनली, कि अगर किसी पेशेंट को हम आइसोटोनिक सोल्यूशन देते हैं तो उससे जो है उसके आरबीसीज के ऊपर कोई इफेक्ट नहीं होता है। लेकिन अगर गलती से या किसी वजह से हम एक्स्ट्रा हाइपोटोनिक सोल्यूशन देते हैं तो वो क्या करेगा कि हाइपोटोनिक सोल्यूशन जो है वो उसके अंदर सोल्यूट्स की कंसंट्रेशन कम होगी, तो वाटर जो है वो मूव करेगा इनसाइड द आरबीसीज, एंड द आरबीसीज विल स्वेल एंड अल्टीमेटली बर्स्ट। सिमिलरली अगर हाइपरटोनिक सॉल्यूशन इंजेक्ट किया जाए तो उसकी वजह से क्या होगा कि वाटर विल मूव फ्रॉम द cells, RBCs, out of the RBCs, and the RBCs will shrink down. Now, after that, of passive diffusion, what is the active transport? Active transport is the movement of substances against the concentration gradient using energy with the help of carrier protein. तो तीन चीजें जो हैं वो एक्टिव ट्रांसपोर्ट में इंपॉर्टेंट हैं कि इट इज अप हिल मूवमेंट, दैट इज मूवमेंट अगेंस्ट द कंसंट्रेशन ग्रेडियंट। सेकेंडली इसमें एनर्जी की रिक्वायरमेंट होती है। और थर्डली कैरियर प्रोटीन की रिक्वायरमेंट होती है। सो अगर डेफिनेशन करें हम तो active transport, it is the movement of substances against the concentration gradient using energy with the help of carrier proteins. अब आगे इसके अंदर जो है वो फर्दर आपके आ जाते हैं, प्राइमरी एक्टिव ट्रांसपोर्ट आ जाती है और सेकेंडरी एक्टिव ट्रांसपोर्ट आ जाती है। नाउ व्हाट इज प्राइमरी एक्टिव ट्रांसपोर्ट? प्राइमरी एक्टिव ट्रांसपोर्ट इज द एक्टिव primary active transport is the active transport of molecules by directly breaking down the ATP molecules for use of energy। ठीक है, तो जो प्राइमरी एक्टिव ट्रांसपोर्ट है इसमें डायरेक्टली ब्रेक डाउन होती है एटीपी की, or carrier proteins are involved, and these carrier protein are called as pumps, and these carrier proteins or pump have the ATPase activity, that is enzymatic activity to break down the adenosine triphosphate to ADP and phosphate, and then which release the energy, and that energy released is used for the uphill movement of the substances. Many of the substances like sodium, potassium, calcium, hydrogen ion, and iodides can be transported by the primary active transport. जो सबसे पिक्युलर एग्जांपल है प्राइमरी एक्टिव ट्रांसपोर्ट की, वो सोडियम पोटेशियम पंप है। वो क्या है? सोडियम पोटेशियम पंप है। तो सोडियम पोटेशियम पंप जो है, इस पे जो कैरियर प्रोटीन है इसके अंदर तीन बाइंडिंग साइट्स होती हैं सोडियम की, विच आर प्रेजेंट इनसाइड द सेल। there are three sodium binding sites which are present inside the cell, and there are two binding sites for the potassium ion which are present outside the cells. So whenever three sodium and two potassium ion bind with this receptor protein, there is a conformational change taking place, and ATPase enzyme will be activated, and ATP is degraded to ADP and phosphate, and energy is released, and sodium is moved uphill against the concentration gradient from inside the cell to the outside, and potassium is moved against the concentration gradient from outside the cell or from the extracellular fluid to the intracellular fluid. Now, what are the functions of sodium potassium pump? Sodium potassium pump का जो main function है, it establish the sodium and potassium concentration gradient between intracellular fluid and extracellular fluid. हमने पहले पढ़ा था कि there is specific concentration of sodium ions and potassium ion in the intracellular fluid and the extracellular fluid, and the establishment of this intracellular fluid and extracellular fluid concentration is maintained by the sodium potassium pump. So it also helps in development of electrical potential across the cell membrane, or it is electrogenic. So there is more electric negativity within the cell, inside the cell as compared to the outside. And it also maintain the cell volume, or sodium potassium help in the regulation of the cell volume. Along with sodium potassium pump, there are other pumps present as well, like calcium ATPase pump and the hydrogen ATPase pump. The calcium ATPase pump is mainly present in the muscle membranes, especially in the sarcoplasmic reticulum, and it concentrates the calcium concentration in the sarcoplasmic reticulum. Hydrogen ATPase pump is present in the tubular cells of the kidneys, especially in the late distal tubules of the kidneys, and also in the parietal cells of the stomach, and they concentrate the hydrogen ion concentration in the parietal cells of the stomach. These we will also discuss in details when we are discussing the concerned sections like muscle and the kidney. Energetics of the primary active transport. The energy required is proportional to the logarithm of the degree that the substance is concentrated. ठीक है, इसके लिए ये एक फार्मूला यूज होता है, ये आपको ज़्यादा बेशक ना पता हो, लेकिन इतना एक फार्मूला पता हो कि द एनर्जी required for the transport of substances across the membrane is equal to 1400 log of concentration inside into the concentration outside. अब सेकेंडरी एक्टिव ट्रांसपोर्ट जो है, दैट इज आल्सो एक्टिव ट्रांसपोर्ट। एक्टिव ट्रांसपोर्ट है तो इसका मतलब है कि जो मूवमेंट है वो अपहिल होगी, दैट इज फ्रॉम द फ्रॉम द लोअर कंसंट्रेशन टू द हायर कंसंट्रेशन। लेकिन इस केस के अंदर एटीपी की डायरेक्ट ब्रेक डाउन नहीं हो रही होती, इसमें एटीपी की डायरेक्ट ब्रेक डाउन नहीं हो रही होती है। दिस ट्रांसपोर्ट इज आल्सो मीडिएटेड बाय द कैरियर प्रोटीन। कैरियर प्रोटीन इन्वॉल्व होते हैं, और कैरियर प्रोटीन की जो है वो एटीपेज एक्टिविटी नहीं होती है, उसके अंदर एटीपेज एक्टिविटी नहीं होती है। और नॉर्मली जो है सोडियम इज जनरली यूटिलाइज फॉर सेकेंडरी एक्टिव ट्रांसपोर्ट ऑफ अदर सब्सटेंसेस, क्योंकि ड्यू टू द सोडियम पोटेशियम पम्प there is higher concentration of sodium ions outside the cell, and now due to higher concentration of sodium ion outside of the cell, it has the tendency to move by diffusion to inside the cell or into the intracellular fluid। अब इसके अंदर ये कैपेबिलिटी पैदा हो जाती है कि ये फ्रॉम आउटसाइड द सेल टू द इनसाइड द सेल मूव कर सके। तो इट कैन बाइंड विद अदर सब्सटेंसेस लाइक ग्लूकोज एंड अमाइनो एसिड, and can also move them into the cell against their concentration gradient. So sodium ion will be moving along its concentration gradient, but it will bind with other molecules and can drag them against their concentration gradient. And when the molecules are moving along the concentration along the sodium ions, that is in the same direction of the sodium ion, that is called as co-transport. And there is also counter transport occurring. There is also counter transport occurring in which sodium ions will be moving inside the cells while another molecules will be moving outside the cell against the concentration gradient, and that movement is called as counter transport. This is the diagram representing the molecule sodium ion and glucose ion are binding together, are binding on the outside side of the receptor, and then they are moving into the cell. Glucose is moving against the concentration gradient and sodium is moving along the concentration gradient. सो को ट्रांसपोर्ट, इसमें क्या है कि सोडियम की हायर कंसंट्रेशन होती है एक्स्ट्रासेलुलर फ्लूइड में, और ये मूव करता है इनटू द सेल अलॉट वेयर द कंसंट्रेशन ऑफ सोडियम इज लो, एंड another substance which has low concentration in the extracellular fluid moves into the intracellular fluid where its concentration is higher, that is an uphill movement, that is a movement against the concentration gradient। काउंटर ट्रांसपोर्ट में क्या है कि उसी तरह सोडियम जो है हायर कंसंट्रेशन से लोअर कंसंट्रेशन में मूव करता है फ्रॉम एक्स्ट्रा सेलुलर फ्लूइड टू द इंट्रा सेलुलर फ्लूइड, और जो दूसरा सब्सटेंस है वो थ्रू द कैरियर प्रोटीन मूव करता है फ्रॉम द इनसाइड द सेल टू द आउटसाइड द सेल अगेंस्ट द कंसंट्रेशन ग्रेडियंट, जैसे कि इस डायग्राम में रिप्रेजेंट किया गया है। अब सेकेंडरी एक्टिव ट्रांसपोर्ट, को ट्रांसपोर्ट की एग्जांपल में सोडियम ग्लूकोज को ट्रांसपोर्ट एंड सोडियम अमाइनो एसिड को ट्रांसपोर्ट रीनल प्रॉक्सिमल ट्यूबुलर सेल्स में होती है, जो कि एग्जांपल है। और काउंटर ट्रांसपोर्ट के अंदर सोडियम कैल्शियम काउंटर ट्रांसपोर्ट, जो कि ऑलमोस्ट ऑल द सेल्स ऑफ द बॉडी में होती है। मोreover सोडियम हाइड्रोजन काउंटर ट्रांसपोर्ट रीनल प्रॉक्सिमल ट्यूबुलर सेल्स के अंदर भी होती है। तो दीज आर द एग्जांपल्स ऑफ द सेकेंडरी एक्टिव ट्रांसपोर्ट। So this is the summary of different processes which are taking place during active and passive diffusion. So here we will finish the lecture for today, and inshallah in the next lecture we will be discussing the vesicular transport, that is endocytosis, exocytosis, and the transcytosis. Hopefully, this lecture will be useful for you. Thank you so much for listening. Jazakallah.